Welcome, everyone. My name is Alka Chaubey, and I'm the Chief Medical Officer of Bionano. And I would like to talk to you today and take you on a journey with me on the new lens of cytogenetics, optical genome mapping in focus. When I talk about optical genome mapping in focus, what I'm essentially referring to is changing the status quo of cytogenomics. So everybody would remember from the 1970s, how did it start? How were the whole genomes being reviewed and looked at using conventional G-banded karyotypes? But now where is it going? It is going towards the next generation level of evaluating structural variations in the human genome using a technique like optical genome mapping. Let me just refresh everybody's memory when it comes to the different tools and techniques that are utilized for interrogating both sequence variants or structural variants in the human genome. If anybody wants to interrogate base pair level changes or look at indels and sequence single nucleotide variants, short read and long read sequencing really performs extremely well at detecting those variants. However, according to different professional medical society recommendations, cytogenetic methods such as karyotyping, FISH, and chromosomal microarray have been the methods as standard of care for evaluating large structural variants. If we look at this genome or chromosome level continuum, what you can see is that every method suffers from different limits of detection. So as you can see, there is a huge coverage gap between the legacy cytogenetic methods and the sequencing methods, both short read and long read, where structural variants in the size range are mostly undetectable. Optical genome mapping really comes in and thrives, not only addressing the limitations of the cytogenetic methods, but also filling this coverage gap and as a perfect complementary tool to either short read sequencing or long read sequencing. Now, why is this really important? If you look at the heme structural variant analysis guidelines by the different organizations such as NCCN, WHO, or NHS, this is just a snapshot of the different region and variants that are recommended by, for example, all three professional societies, or the two of them, or one of them. Moreover, all of these professional societies routinely update their recommendations and guidelines, and hence it becomes extremely unmanageable for laboratories to keep adding different targets to their test menu to interrogate these regions of the genome for a specific heme indication. So optical genome mapping, correctly identified as the new lens of cytogenomics or cytogenetics. And I want to give credit to Dr. Alexander Heuschen for this slide, where he referred to optical genome mapping as high resolution digital karyotyping. If you look at the karyotype on the left-hand side, each band is roughly 5 megabases in size in a constitutional application, or roughly 30 to 40 megabase in size in a cancer application. And on the right-hand side, what you see is these ultra-high molecular weight DNA molecules. And these specks in green are similar to a very high resolution barcode, where optical genome mapping allows for the detection of chromosomal aberrations as small as 500 base pairs and as low as 5% allele frequency, thereby providing almost a 10,000-fold resolution. Not only that, irrespective of which sample types you start your assay with, in our software provided by Bionano, which is called the VIA software, which stands for Variant Intelligence Applications, this is a software that allows for the integrated solution where in a single platform, different methods or techniques such as OGM, NGS, and microarray from the same sample can be visualized, interpreted, and then reported, thereby maximizing meaningful insights from these different methodologies. Now, I have to give you an example of acute myelogenous leukemia, where optical genome mapping lens allows for the detection of cryptic or pathogenic structural variants. So on the left-hand side, you see this karyotype where a gain of chromosome 8 and chromosome 19 are identified very easily. However, on the same sample, when optical genome mapping was run, not only were the gains identified, however, a cryptic and a pathogenic KMT2A MLLT10 fusion was beautifully detected. And this, again, this case is courtesy from Dr. Adam Smith when he was at University Health Network in Canada. Not only that, when you look at complex karyotypes with the OGM lens, you really evolve and elevate your experience from subjectivity to objectivity for informed decision making. Again, on the left-hand side, all the cytogeneticists and pathologists would be familiar with this sort of karyotype, giving this absolutely complicated ISCN nomenclature where you have an add, you have a question mark, you have a marker, you have a composite karyotype. It becomes extremely difficult to make sense of this, except, yes, it is a complex karyotype. Now, when you run OGM on this case, not only do you see the complex karyotype, but you also now have evidence of which oncogenes are part of these structural rearrangements so that you have more precise details of these abnormalities present in the sample. Now I want to show you a couple of examples. So Dr. Gwilyn Tang and her team from MD Anderson Cancer Center published recently the first use of OGM to detect chromoanagenesis, a key marker of poor prognosis in AML. Now, this publication is extremely important because it allows for the accurate assessment of chromoanagenesis in 16% of samples and which correlated with highly complex karyotypes, monosomal karyotypes, and TP53 mutations. Not only that, however, the correct detection of chromoanagenesis was also evaluated with respect to a median survival for AML subjects, which was only 5 months as compared to 14 months in individuals without chromoanagenesis. This also allowed for the refinement of the risk stratification, where optical genome mapping's high resolution and sensitivity provided an alternative to redefine AML risk stratification to potentially guide treatment planning in this research. We also have another very important publication from France, where this group of researchers evaluated OGM as an effective method to analyze multiple myeloma samples. Now, we all know that multiple myeloma is a plasma cell myeloma malignancy where limited sample quantity is a huge problem to evaluate and detect large structural variations in this malignancy. So this group of researchers used a novel OGM workflow where they mixed CD138 enriched and unenriched cells, and they were able to reduce the input requirement of these cells by 50%, which allowed them to have OGM concordance with FISH as high as 93%. However, they also detected 22 variants which were missed by FISH, but which were extremely relevant for prognostic risk evaluation. I encourage all of you to go through and review this publication, which was published in the Journal of Molecular Diagnostics early this year. Finally, I want to show to you a timeline of the evolution of optical genome mapping for human clinical research applications. On the top, you see the timeline along with the publications that really were landmark publications on large data sets, multi-site, multi-operator, multi-instrument, clinical trials, both in postnatal and prenatal applications in 2022. This was followed by ISCN getting published in 2023, and we hit the 1,000 publication and 5,000 human genome mark last year. If you look at the first validated protocol for FSHD using optical genome mapping. That was in 2019. And as of 2025, we have over 200 validated protocols. But there are a number of milestones that were achieved in this with the community. And you can see all of this. I touched a little bit upon the International Heme Framework publication, but also the French cytogenetic guidelines were issued in 2023 recommending optical genome mapping as an alternative to cytogenetic methods. In the United States specifically, the American Medical Association has established now 2 Category 1 CPT codes for optical genome mapping, one for hematological malignancies, and there is one in progress for constitutional disorders. But we have also crossed over 10,000 human genomes published using optical genome mapping. And we also have a huge initiative driven by ACMG, AMP, CAP, a points to consider, which is in the works. So stay tuned for more progress coming from the community. Now, finally, what's new? What's coming? So we have just released the VIA 7.2 software and compute platforms. And for those of you that are evaluating upgrading to this software, what we have actually provided is using this VIA 7.2, you can transform the workflow of OGM in constitutional genetic disorders. But also the significance associated with phenotype scoring will now be available for OGM data to help streamline interpretation of OGM variants. So a lot more of these improvements will allow, especially on our Stratus compute, and our software upgrades will allow for users to double the number of cancer samples per week. And there are significant improvements in our underlying algorithms that allow for the better detection and improvements in sensitivity, specificity, and resolution for SV detection. So with that, I'd like to thank you for your attention, and please feel free to reach out to me if you have any questions. Thank you.